Hey guys, Mr. Gomes here. So today we are going to learn how to do a chromatography lab. Here we go. Okay, so for my chromatography lab, what I'm doing is I'm going to try to separate a mixture, or at least something that I think is a mixture, so I can get the different pigments in that mixture and see if it actually is a mixture, and if it is a mixture, what the different pigments are that are in that mixture. Okay, so we're going to use a few different things. We've got chromatography paper. I got a little piece here left over. I'm going to check the ink of a marker. I've got scissors to cut stuff up. I've got wooden splint. I've got some tape. I'm also going to do it with some spinach leaves, so we'll see that in a second. And I've got an Erlenmeyer flask. Okay, so you see this kind of somewhat triangular shaped beaker thing. It's called an Erlenmeyer flask. Okay, so we will be using these to set up our chromatography lab. So the first thing we want to do is to cut the chromatography paper to the measurements in your instructions, and you're going to get a strip that looks like this. Okay, so the next thing I'm going to do is I am going to draw a pencil line right here. Okay, so I can know where to put my dot for my chromatography. All right. Now you can see there where I drew the line. Okay, I didn't draw a perfect line. I'm not good at those things. But you can see there where I drew the line for the beginning of where I'm going to put my dot for my marker. Okay? Now I'm going to go ahead and carefully place a dot right there. Okay? Now I have two examples. Okay? This example right here is a great example. I know it's a little bit blurry, but it shows just the one dot on the line. So when you get the marker, you literally just go boop on the line. This is a bad example. It's the one you saw me recording earlier. Okay, this is what I don't want you to do. Okay, because this is going to make the dye, the ink separate into the dyes in a not very nice way, and we want it to look good. Okay, so that's how you're supposed to do that, just one dot. The next step is to get the wooden splint, and I'm going to tape my chromatography paper with a little piece of tape there to my wooden splint. Okay, now I am going to put enough water in my Erlenmeyer flask so that when I put this in, okay, when I put the chromatography paper in, this tip here, that's why it's cut like this, this little tip is going to, let me show you right there, this little tip is going to touch the water. We want the water level to be below the dot, because if the water level is above the dot, that's going to mess up the whole thing. So I'm going to put just enough water there in my Erlenmeyer flask to make sure that it touches the tip, okay, which in my case it did not. So I'm going to take it out and just fill it up with a little bit more water. Now what you see there is that very tip of the chromatography paper is touching the water, and you probably can't see it very well, but the water is slowly traveling up that chromatography paper. And as it travels up, it's going to take the ink with it, and it's going to separate the dyes based on how much each dye likes the chromatography paper. Now that I have my ink with the chromatography paper set up, I'm going to do the one with the spinach next. Now, this may be difficult to see, but I drew a pencil line right here, and then I got a spinach leaf, I got a penny, and I pressed the spinach leaf right above that pencil line. And what is there, what I can see with my naked eye, I don't know if you can see it or not, is that green juice that comes out of the spinach. But I did it very slowly, very firmly, and kind of uniformly. And what I got was just a nice solid green line all across that pencil line. Now I'm going to do the same exact setup as my ink. Okay, the only difference is I'm going to use a different solvent called the chromatography solvent. So I'm going to set that up and then show it to you. Now what I have set up here is my spinach leaf juice right there. Okay, and here's the chromatography solvent. It looks exactly like water, but it is not. Okay, it's a mixture of something called petroleum ether and acetone, just because the chlorophyll in that spinach juice is going to have different properties. And what we're going to do here is we're going to split up the different chlorophylls, okay, in spinach. Now notice how I have this like waxy film coating on top of the Erlenmeyer flask. It's because that chromatography solvent right there does not smell nice. And we don't want to be smelling that for a long period of time, so we're going to make sure that that stays covered while the chromatograph runs. All right, and then we're going to come back and look at the results. Okay, so what we have here are the results. I just took a picture so it would be easy to see them. So here is what happens when the orange marker gets that dye, the ink gets separated into the different dyes, okay? So I don't know how well you can see this, but there's definitely a yellow dye that makes that up. And there's like a dark red and then maybe a lighter red or a pinkish dye. Okay, so there's 3 different dyes that make up that orange ink color in the marker, so we definitely know that that's a mixture because we can see the different parts. Now, one thing I want to point out is, I don't know how well you can see it, but right here, okay, you can see where the chromatography paper was wet and where it's dry. So it's wet down here and dry over there. So what that means is this, okay? The dye within that marker ink that likes the water the best will be the closest to the water. So this pinkish color over here is the one that likes the water the best. Okay, and the yellow one down here is the one that likes the water the least. So that means that there's something about the molecular structure of that pink dye that's similar, more similar to water than the yellow dye. Okay, now let's look at the spinach. So here is a picture of the result from the spinach. Okay, so here is the line where we started, where we had that spinach juice. And then the spinach traveled upward, okay? And I don't know how well you can see that, but there's 4 distinct colors. There is an olive greenish dye, so to speak, over here. There's a darker green. And then there's a really thin light yellow line, and then there's a definite darker yellow line. And we can measure their distances that they traveled from the original line, okay? Now, interestingly, for this one, the dark yellow and the solvent, the chromatography solvent, traveled perfectly together. Okay, so all the way up. So that means that that dark yellow pigment and the solvent are very similar to each other. And then the olive green here is a very different, or perhaps not say very different, but the most different of all the pigments that we got to separate. The olive green is the most different from the solvent, okay? Now, each of these pigments here that we're separating are actually different types of chlorophylls that are in spinach, okay? So the colors that you see in the fall, you know, from trees, typically people don't look at spinach in the fall, but when the leaves are changing colors, because the green chlorophyll dies first, but then there is these other auxiliary or supporting chlorophylls that bring out the different kind of orangey, yellowish, reddish tones that we see in trees in the fall. And you get to see 2 of those tones right here, the light yellow and the dark yellow. And that's it for a chromatography lab.